Hello everyone, I'm Qiyang Shou from University of Southern California. In this presentation, I'm going to talk about my work of total generalized variation TGV constraint reconstruction improves test-retest reliability of high-resolution 3D pCASL in children. I have nothing to disclose. Abnormalities in regional CBF in developing population may lead to some developmental disorders, such as autism spectrum disorder, adolescent depression, ADHD, or substance abuse. ASL or arterial spin labeling is a useful tool for studying CBF as it's completely non-invasive and it is able to quantify absolute CBF values. However, since ASL usually has low SNR, which means it is usually acquired with low resolution, that prevents from analysis for small brain structures. And also, the relative long scan time makes it especially difficult for young subjects as they tend to have more head motion during the scan. So the goal of this study is to develop a high resolution, which is 2 mm isotropic, single-shot multi-delay ASL acquisition scheme with 8 times undersampling and TGV reconstruction. And we evaluate the repeatability of this method on pediatric subjects with test and retest scans. In our study, we used the 3D GRACE readout with a Kuiper-Reynolds undersampling pattern. Each PLD we have 8 measurements. The acceleration factor is 2 in the KY direction and 4 in the KZ direction. As you can see, there is a shift of the pattern for each measurement, so that we can get a complete K-space if we combine all 8 segments together. And this combined K-space can be used to estimate sensitivity maps for reconstruction, so we do not need to acquire a separate fully sampled reference. For the image reconstruction, we use the constraint method with TGV regularizer. From this equation, you can see that it regulates both control and label images, as well as the perfusion image, which is denoted by C-L in the last term. After this reconstruction, we can get all 8 control and label images, as well as the denoised perfusion image. This slide describes the imaging protocol for our experiments. We used the 5-delay pCASL sequence with the previous undersampling pattern. Background suppression was applied and optimized for each PLD, and we used a high-resolution acquisition 2 mm isotropic. Our matrix size is 96 by 96 by 48, so our total coverage will be 96 centimeters, and our total scan time is 8 minutes and 33 seconds. We scanned 19 young subjects aging from 9 to 17 years old with test and retest scans that are 2 weeks apart. We evaluate this method by first calculating the SNR of the perfusion image for each PLD, and also the SNR for the fitted CBF and ATT maps. And finally, we calculate the ICC of test-retest in CBF and ATT values. We compare 2 methods. One is the combined method, which means we combine all the segments of K-space to get 1 perfusion image for each PLD. And we also compare the reconstructed method, which means we reconstructed each perfusion image and average all 8 perfusion images for each PLD. Here are some results. This figure shows the combined and TGV reconstructed perfusion images for the 5 PLDs. We can see that the perfusion image with TGV reconstruction have higher SNR compared to the combined method in all 5 PLDs. The table shows the quantitative SNR measured in the gray matter ROIs. And in all 5 PLDs, TGV reconstruction can improve SNR by about 30%. And we also get the CBF and ATT maps with the 5 PLD images by fitting the kinetic model. This figure shows the CBF and ATT maps from the 2 methods. The CBF and ATT maps from the TGV reconstruction have higher SNR and gray and white matter contrast compared to the combined method. This figure shows the scatter plots of the test-retest performance of global CBF and ATT for both methods. We can see that the TGV reconstruction improved the ICC from 0.46 to 0.65 for the global CBF, while ICC for the ATT was comparable for both methods. We also did a regression study to see the trend of CBF and ATT with age. We found that CBF had a decrease with age in this young age group, while the ATT does not significantly change with age, which is consistent with previous studies. In this study, we achieved the 2 mm isotropic 3D single-shot 5-delay pCASL within 9 minutes. We see that TGV reconstruction can improve SNR for the reconstruction perfusion images for all PLDs, and with the improved SNR, we can get better repeatability of CBF quantification. In conclusion, we achieved an ICC of 0.65 for global CBF, which is consistent with previous studies in children, and much higher resolution, and we also achieved the 5 PLDs. This shows the potential to use multi-delay ASL for neurodevelopment studies. Thanks for your watching.